Protein separations, yes. And then after we separated, we created a protein that we're going to use. We used a rabbit. So what were we making inside that rabbit?
Antibodies.
An antibody. What is important about an antibody?
That's off antigens.
It does fight off antigens. That's in the normal biological sense. But what do we get with antibodies for laboratory use?
A lot of them.
We want to get a lot of them, yes. What is the characteristics of an antibody that makes it important for us to use? It binds specifically. It will bind one and only one thing in a mixture of things. It binds specifically. It also binds tightly, meaning this, that if any of the antigen is in your mixture, it will bind at very low concentrations. So you can be very specific. So tightly and specifically, we can detect whatever we're looking for in a mixture of things. All right? And so we can create antibodies using a biological system, something that is already designed to create antibodies. And so we use an animal system to do that. And the antibody that we're looking for, the version, is the IgG, the class. It is the most soluble and most often produced antibody. And so as long as the animal is producing it, we continue to take a little bit of blood, isolate that antibody, and that animal will consistently make that antibody for its lifetime. All right, it will be immunized against whatever it is and create those antibodies. All right, we can isolate those antibodies and then use them. What we first created are called polyclonal antibodies. Okay, what in the world is polyclonal? All right, remember you have an antigen, so some sort of molecule that elicits an immune response. And then an antibody could bind right here. Oops. An antibody could bind right there. Let me do that in a different color. Let's do it in green. You guys can see green. Okay, where the antibody binds, what is that called? That is called the epitope. Okay, now there are many places on the antigen in which antibodies bind in normal, when it's created normally in a rabbit or a rat. And you will have lots of antibodies that bind, but only one antibody binds at that epitope, a different antibody binds at this epitope, because it's a specific shape on that particular antigen that it's binding to. And so you could bind 5 or 6 different antibodies to your antigen in 5 or 6 different places. Okay? So, in terms of immunity, having antibodies bind something initiates the immune response to get rid of it. That's the natural process. That's good. We want to cover something that's not supposed to be there with antibodies to get rid of it. Okay? Now, if in the laboratory we just want to use it as a tool to bind to the protein, then binding multiple things could be good in your sample, okay, so that we make sure that we bind to it. But this is what's called a polyclonal. It's a collection of antibodies that bind your one antigen, but on different epitopes. Okay? And so, that's great, but may not be exactly the same thing that you want for some other experiment. So we're going to create a different antibody. We're going to create what's called a monoclonal antibody. Now, a monoclonal is creating one antibody that binds at one epitope on one antigen. So you get 1-to-1 ratio binding. This allows us to be quantitative rather than qualitative. Okay? Quantitative, 1-to-1. If more bind to the antigen, there's more antigen there because I only have 1-to-1 binding. Polyclonal, though, tends to be not as quantitative, but it is qualitative. Is it there or not? Yes or no? But you can have multiple bindings to one antigen and a few bindings to another antigen, so it's a little bit less quantitative, not one-to-one relationship. So we want to create a monoclonal antibody that has one specific binding to one specific epitope. Again, we will use a biological system, an animal, to produce the antibodies. Instead of getting the mobile or the soluble antibodies, we will actually isolate the beta cells that are producing the antibodies. All right. Now, though, here's the problem. If we isolate a cell has a lifetime, and so we could grow that cell and we can collect the antibodies that are being produced, but over time that cell will die because it has a certain lifetime. But what we will do then is take the beta cells that are being producing the antibodies and we'll fuse them to immortal tumor cells, a cancerless line. And we will make a mixture in which the cell will produce antibodies, but it is now immortal. And so as long as you feed it, it will continue to grow. And it will grow rapidly, and it will produce lots of antibodies. All right. If I take the beta cells, a collection of beta cells, I'm going to create lots and lots of different antibodies. Okay? And so at this point in time, it is still polyclonal. I'm making lots of different antibodies for lots of different epitopes. But what you can do is grow them on techniques that you learn in cell and molecular biology or in micro. You grow them on the plate and you collect individual cells, let them grow as a colony, and if you can isolate one cell that produces one antibody, allow that to grow as in a colony, just collections of itself, you can isolate one particular antibody, and we will then have monoclonal antibodies, samples of those. Okay? And this is a wonderful thing. We don't have to keep an animal alive. We just have to grow up cells. Those are fairly easy to do, and we can continually make that antibody. All right. So how in the world would we use an antibody? I'll show you several techniques by which this is not exclusive of all the ways that we use antibodies, but these are some common techniques. An ELISA is a test, an enzyme-linked immunosorbent assay. All right, well, where in the world did the antibody come from? The immuno part. Okay, we're going to use an antibody as part of this. Okay, but there's a whole lot of other things. Sorbent, we're going to absorb proteins onto a plastic well. Assay, we're going to test how much is there. And enzyme is going to be a part of the process. Okay, and so let me show you the animation. Again, this will be linked on Moodle. Okay, so the second is more often used, the second type. So what you do is you have a well, okay, and the well has nothing in it, but that well is a plastic container that will absorb protein, okay? Antibodies are protein and your sample is protein. So what you do is you put your sample inside the well, okay? So now your well is full of sample and proteins will adhere to the edges. Now, those proteins will be everything in the mix. Some of those proteins might be the one you're looking for. Some of them may not be what you're looking for. All right, but there's still protein floating around in the solution. So what we do is we drain it and we wash it. Okay, so wash. Now, you've got protein that is on the edges. Well, here's the problem. The protein sample that you put in doesn't actually block up all of the plastic. So what we have to do is one more step called we have to block it. Blocking is putting a very high concentrated protein that we know is not part of our test. We use two things. We use BSA, bovine serum albumin, which is a very common standard protein, or we use milk, powdered milk. And the milk will coat every other part and it will make sure that there's no plastic that is open and available. So we block it. So now we have our protein and then all these other proteins that we know don't react. Okay. What you will do though is in one well, you will not put your sample in, but you will block it so that you have a test to make sure that that is a negative control, that the protein that you use to block isn't causing. Next, you put in a solution of your antibody, the one that is the one designed to bind to your protein. All right. In this case, it may or may not bind to something, depending upon what is there. Okay. Hopefully, if you set up the experiment right and the protein that you hope is there, it should be there. All right. At this moment in time, you've got your antibody in that mix. You need to wash it again. Okay. Then you may have something like this: your protein and your antibody bound. Okay. Washing, make sure that the binding is specific. It is not just an antibody that's adhered to it and kind of stuck to the side. You wash it, and only things that are actually doing a physical binding will stay. Everything else gets washed out. The next thing that you do is you put in what's called a secondary antibody. Normally the secondary antibody is this: if you used an antibody made from rabbit, use an antibody that is anti-rabbit. It will bind to every rabbit antibody ever made. And you put it in, and you get a secondary antibody binding. But the secondary antibody is bound to an enzyme. Okay. And so, the last step after you wash is to add in a reagent. A reagent that is placed in the solution that is no color, but if the enzyme is there, it will change it in color to produce something you see. So, what you measure is the amount of the resulting color. If you have a lot of this, that meant you had a lot of enzyme. The only way you have enzyme is if you have the secondary antibody. You only have the secondary antibody if there's a lot of primary antibody on there. And if there's only primary antibody there, if your sample is there, if you have washed it well and blocked it well, and you've done this. So you do have to have controls to make sure that your washing and your process works, where you have a control where you don't put in your antigen, you put everything else. That should be absolutely clear, no color. And you want to put a control in where you put the known protein that you're looking for, so you know it's there, and you should produce color. If you get one control that says no color when it's not supposed to be there, and color where you're supposed to be there, then anything in that experiment, you can trust a little bit that the data is good. What you're going to see is a set of wells, and a lot of times we use a well plate that will read, and the well plate reader will do it very quickly, but you can see each sample has a different amount of color. All right, so you're making all of this sandwich and you're doing all of these processes, and the final product, we're actually looking at an organic molecule creating color that represents that your protein was actually there. An example of an ELISA that is used, you can go to the supermarket and you can buy one, and you put the sample on it, and you either get 2 lines or a plus line, or no 2 lines and no plus line. Basically what it is looking for in a pregnancy test, it's an ELISA reaction that is looking for a certain protein that is produced when you are pregnant. And it's looking for that particular protein with an ELISA exam. Okay? A lot of times this is a very powerful technique to do your experiment. The reason why we do this one, that antibody is expensive. This antibody is cheap. Okay? And so in the first example in that animation, you actually had to have 2 antibodies that bound your antigen. That's 2 expensive antibodies. How expensive is this one? I used 1 antibody in grad school. I got 100 micrograms of the antibody for $500, which I could do 5 experiments with. And the secondary antibody, I bought a bottle that would last me 6 months for $50. And so sometimes antibodies are the expensive part of your experiment. All right, a lot of times you will see this Western blotting technique in experiments, and basically on the journal article, all you're going to see is this little box with these little dark spots. That's a Western. A Western blot starts with this. I'm going to separate proteins. Okay, so you do your experiment, you do different variations of the experiment, and every lane will represent a different variation of what your experiment does. You will separate it by size. You will then have all the proteins in that jello, but in the jello it's hard to deal with. So what you do is you put a piece of paper, okay, nitrocellulose or their other filter membranes here, and then you put your piece of jello, the gel, on top of it. Then you change the electricity. You put a positive electrode here and a negative electrode, a positive electrode here and a negative electrode there, and you force all the proteins to move towards the paper, and they get stuck on the paper. So you get an impression of every protein that was there. All right? You only mix up your electrodes once, all right? And because if you go the other direction, you've lost your entire experiment. All right, take off your jello. Now you have a piece of paper that has all your proteins on there. That's nice because then you can put it in a solution of, what you do is you block it by making sure all of that paper is filled with protein that doesn't react. You wash it, and then you put it in a solution of primary antibody. The primary antibody will bind, just like the ELISA, and then you wash it. You put a secondary antibody with a enzyme, and then you give it a reagent. It can be colored. It can be chemiluminescent that will shine light. It can be a fluorescent dye. There's all kinds of things that we now have to get very sensitive types of responses. And if the protein of interest that you're looking for is there, the antibody will bind to it, and then you will get some sort of signal from it. Okay. In this case, again, the sample could have hundreds of proteins. You're looking for one. Is it there or is it not? And is it there in high concentration? Is it there in low concentration? What you're going to be comparing is the different experimental conditions. If set up right, the amounts that are there will give you a clue as to the question you're trying to answer. And so when you use these blots, you're always comparing against, you should have some controls, a positive control, a negative control to make sure everything's working, and experimental conditions that change the experiment to see, answering the question, and what the difference in those blots or those lanes read. And that's how you read a Western blot. The last that we're going to introduce is immunoprecipitation. A lot of things that we need to know in biological systems is how things are interacting. And so, by chance, your protein of interest does a certain thing, whatever that biological process is. But that protein very rarely does things out of context or by itself. Usually there are other things that work with that protein to do a larger process. Okay? But sometimes we don't know exactly what those other pieces are that are working together. And so what you can do is this. You can investigate what proteins bind together, and if they bind together, that means they probably work together using immunoprecipitation. So what you do is you put your protein of interest into the mix, all right, and you allow it to float with all the other proteins. Okay? And if they bind together, they're going to form a complex. So you give it time to form the complex. You then introduce an antibody to the one protein that you are interested in. That antibody binds to your protein very tightly. Okay, so now you have the complex of how it normally binds to other things, and now you got your antibody bound to it. There are techniques to separate out antibodies. So what we do is we use column purification and will pick out antibodies. All other proteins flow out through the column. If you wash it, everything gets washed out except for binding to the antibody, which is bound to your protein. If that protein binds to anything else, they'll be attached, even through the washing stages. You release the antibody. You now have a collection of at least the protein that you've trapped and anything else that might be there. You don't know if there's anything there at that moment in time. And then you put it on an SDS page gel, and you see how many bands there are. I have precipitated the collection by using the antibody. I haven't precipitated by making it salt out. I precipitated it by using the antibody, by using that to pull it out of solution. And so we can then separate them. We can digest them into smaller pieces. We can do Edmond degradation, or we can do mass spec. Mass spec is another technique in which we can do sequencing, because each individual mass ratio can be associated to different amino acid groupings. It takes a lot more programming to do, but we can isolate and identify what proteins bind together. So those are some ways in which we use antibodies. We will use antibodies in a lot of other ways as we introduce other concepts, but these are some general ones. All right. Next chapter, presentations, protein function. Check this out. We have 8 chapters to cover. We're starting 6. That could be scary. We're way past halfway point now. All right. So all of this study, and this is kind of where biochemistry gets into biochemistry, we've isolated proteins. Okay, so lots of people have isolated lots of proteins up to this point in time. Why in the world do we isolate a protein? Why do we spend all this time isolating these proteins? What do we want to know? How they function. What do they do? Okay, all right, so chapter 6 is going to talk about protein functions. What do they do and how do they do it? How many proteins are in the body? Different individual proteins. Lots. Okay, at one point in time before we did the Human Genome Project, we thought there were about 100,000. We were very surprised when we found out there were somewhere between 20 and 30,000 different proteins in our body that make us do everything that we do. All right. How many do we understand? Not all of them. Okay, we're still looking at. So, how in the world does a biochemist in a textbook teach you every single protein in the body? We don't. Okay? We just can't. So what we do is we give you examples of classifications of types of proteins. Okay? A lot of the different techniques in terms of how they function will translate from protein to protein to protein. Okay? And so you pick up the general ideas that can be applied to other proteins, but you have to know each one specifically. Which ones do we choose to show you? Okay, we're going to show you a handful. We can't show you all. Okay, we can't even show you 100. Right? So we show you 10 maybe. We show you the ones we studied the most and we know the most about. Okay, and from where our understanding has been derived. So a lot of the studies that are presented in books are the ones that are just the best known. We have the most data about. But when you go into your own research, you start looking at your individual protein and how it works. And so here are some examples. Protein functions in lots of different ways. We have from structural proteins to proteins that transport things between the membranes, to proteins that move things around, to enzymes that will facilitate an organic reaction, to proteins that will bind to something to sense that it's there and send a signal in a different pathway. We will have proteins that will organize structure. And so we have lots of different types of proteins. The book puts them into 5 categories: metabolic enzymes, structural, transport, cell signaling, and genomic caretakers. Those are the way that it classifies the proteins. We're first going to find a binding protein, okay? And so when we talk about a binding protein, we have to talk about what it binds. And so we can talk about a binding entity as a ligand, okay? Something that binds and is held on by a protein and then released at some point in time. That general term is called a ligand. When we get to a specific enzyme, the thing that it binds is called a substrate. Another word for that is reactant when we go all the way back to general chemistry. And so a ligand is something that binds and is released. All right. The binding site within the protein has to be a complement to that ligand. And so its shape, its polarness, its charge, its hydrophobicity will be optimized to bind that binding molecule, whatever it is. And it creates an affinity. It can have a low affinity or very high affinity for that particular binding ligand. A low affinity would mean I have to have a very high concentration in the solution before I effectively bind. Okay? A very high affinity means that even a little tiny bit in the solution can bind. And so every protein that binds has a measure of how much affinity it has, or binding constant. The binding of a biological entity is not constant. There is a rate of dissociation for every binding event. And so a protein binds it, holds onto it, and releases it, and then could bind it again and release it, but it is a dynamic state. Bind and release, bind and release, bind and release. The two proteins that are the typical example are hemoglobin and myoglobin. All right, both of these proteins were some of the first proteins to have their masses accurately determined. They're characterized by ultracentrifugation. These are one protein in which a specific biological function was associated to it. These were some of the proteins that had their first X-ray crystal structures determined. And for hemoglobin, this was one of the first examples in which a genetic disease was linked to. So one change in the DNA linked to a disease. And so sickle cell anemia, we'll talk about that. And so we know a lot about this system. Now, they bind to something, hemoglobin and myoglobin. What is the binding ligand in this case? Oxygen. That is not the only binding ligand, though. Heme. This protein, both hemoglobin and myoglobin, cannot function by itself only amino acids. It has to have a functional extra piece. The extra piece is the heme group, called a prosthetic group or a cofactor. Okay? It's non-polypeptide, but it provides biological activity. Quite often, cofactors are derivatives of vitamins. Okay, this one isn't, but a lot of times a cofactor that is used by a protein is from a vitamin. Heme binds two molecules very tightly, oxygen and carbon monoxide. Okay? Both have similar structures. They are linear, and they really look very similar. Okay, which one does heme want to bind to more? Carbon monoxide. Heme by itself in a solution will bind carbon monoxide like I believe 10,000 times better than oxygen. Okay? That's not necessarily good for us. Okay? But it doesn't do that in the protein itself. So it can bind oxygen, but it really, really wants to bind carbon monoxide. Myoglobin, all right, is a polypeptide chain that contains 75% alpha helix in 8 segments. 4 segments are ended by a proline. Proline doesn't like to be in alpha helix and tends to be the protein, the amino acid that ends a alpha helix. If you take a look at the interior of the protein, all of them are hydrophobic with the exception of 2 histidines. So thermodynamically, the histidine is polar and can be charged, are sitting inside the protein exactly where I told you they shouldn't be. Okay? So this causes a little bit of instability in terms of its full, if it really wanted to be totally stable. So when you put polar things inside a place that it doesn't really want to be and it causes a little bit of instability, that probably indicates they are very important to the overall function. And we will see how those two histidines work together to cause the function of both myoglobin and hemoglobin. In this case, those two histidines are interacting with the heme, one of which is actually covalently linked to the iron of the heme. The heme is held together in that the iron is held together by 4 nitrogens in the plane of the ring, but because of the oxidation state of iron, it can have 2 more binding places, one in front and one behind. All right, if you think about heme's ability to bind 4 nitrogens, one in front, one in back, something in front, something in the back, what would be the electron pair geometry of the iron binding to those entities? Because the next thing that comes in is oxygen here to create a binding. If I had oxygen bound, I had the histidine bound, and I had the 4 nitrogens, what's the electron pair geometry of the iron? What is it? Octahedral. And the hybridization of that would be sp3d2. Good. Wow, this stuff never leaves. Right? And so it is an octahedral coordination. Only in one of the oxidation states of the iron. If you change the oxidation state, you change how many coordinations you have, and you disrupt the function of hemoglobin. And so it's important to keep the oxidation state of the iron available to have 6 binding coordinated, 6 coordinations. All right. Now, I told you 2 histidines were important. Now, the histidine F8 and the histidine E7, well, what in the world is that? What is F8 and E7? How many alpha helix are in myoglobin? 8. They are labeled A, followed by B, followed by C, and so it is the number coordinate to the alpha helix, and then the number in that is the F helix, the 8th residue, the E helix, the 7th residue of that alpha helix. So that's where those numbers come in. All right, the histidine is very important. The histidine F8, the one that is behind it, okay. They actually call it the proximal one that's in front of it. The coordinated nitrogen electron pair coordinates and makes a bond to the iron. The 6th position is the distal, okay, and the oxygen comes in and binds. But here is where the histidine gets into play. The 8 is actually linked. The 7 doesn't actually make a bond to the heme. It actually doesn't even make a bond to the oxygen. What it does is it is in the way. It causes function because it sterically hinders. Okay? Well, what in the world does it sterically hinder? Oxygen tries to get in here. Oxygen wants to line up straight, okay, and make a wonderful bonding to the iron. But because this histidine is there, this oxygen gets too close and they repel. So what happens is the oxygen has to bind in at an angle. Instead of going 90 degrees on, it is a 121 degree bind. Why is that important? If you overlap the electrons directly in a 90 degree bonding, oxygen would bind stronger to the iron. It would actually bind so strong that when it binds to the iron in the lungs And when you get down to the big toe where oxygen needs to be, if it bound directly straight, it wouldn't let go. And so it would not be a transporter if it bound that way. It would just bind it and never let it go. So that physical blocking lowers the affinity of oxygen so that it picks up in the lungs, but when it gets out into the extremities, it can let it go. And it lowers the affinity for oxygen, so it's useful to us. Okay? When oxygen does bind, okay, we get, we get a little bit of change in the structure. Stop. Okay. Stop it. All right. Show the cheat. All right. It's giving me fits. All right. Try it one more time. Good. All right. It's moving. All right. Do you see as the oxygen comes in, it's not as smooth as I like it. Ooh, there. Okay, there it goes. The oxygen binds to that iron, and when the oxygen binds to the iron, the protein changes just a little bit. Do you see a little bit of change? A little bit of shift? Okay. There are two forms of this protein. One is the deoxy form, and one is the oxy form. The change in the protein is very subtle, but the affinity for oxygen, it is dramatically different between the two. What causes this change is as the oxygen binds to the iron gets pulled forward. When the iron gets pulled forward, it pulls that histidine that was attached to it. That's attached to the F-alpha helix. Well, when I pull the F-alpha helix, I twist it around a little bit, which twists the entire protein just slightly. And you can see just a couple changes in that. But for that change to happen, I have to break a lot of non-covalent bonds in the entire protein to cause that to shift. And so, because everything is connected, when I pull the string in one edge, it moves their entire, I just lost. So the bit interaction makes the oxygen bind a little less tightly, enough so that it can be bound in the lungs and then let it go when it gets in the extremities. That's in the hemoglobin model. Myoglobin actually has a very, very high binding rate for oxygen. Myoglobin's function is a little bit different. Myoglobin is found in muscle, and it doesn't transport through the bloodstream, not like hemoglobin. Myoglobin's job is to do this. It is to hold oxygen very, very tightly. It will only let go oxygen when the pressure of oxygen in your muscle goes way, way down under extreme exercise. Then it will let go a reserve oxygen so that you can keep your metabolism in an aerobic process rather than an anaerobic process. All right. The polypeptide also surrounds that heme to make sure that it keeps in the +2 oxidation state so it can coordinate 6 things rather than the +3, which would only have 5 coordinations. And so that would change the entire function of hemoglobin. So the protein around the heme lowers the affinity for heme to oxygen. It allows the binding and prevents the oxidation of heme so that it will bind. Also, it will prevent carbon monoxide from binding as well. And actually, it affects carbon monoxide even more than it does oxygen. So it lowers its affinity for carbon monoxide. Quite frankly, it wasn't 10,000, it was 20,000 times stronger. The carbon dioxide versus oxygen, if you just gave them a competition, just heme without the protein. But in that bent conformation, it goes down to 200 times stronger. All right. Here's the key. Here's the key to this. All right. The difference between 20,000 and 200 is 100 times. All right. Here's the deal. Right now, every one of us have 1% of all of our hemoglobin bound to carbon monoxide. Okay. The concentration of carbon monoxide is very, very low. But he loves to bind to it very, very tightly. So very low concentration will actually bind. So we have 99% of all of our heme available to bind to oxygen. All right. If the protein wasn't there, if you have 1% now, and you increase the percentage of what it wants to bind from 200% to 20,000%, if the protein wasn't there, the concentration of carbon monoxide in this air would be 100% bound to hemoglobin, okay, if there's no protein. And so that is very important to us. Also, you do not need to have a lot of carbon monoxide to start to displace oxygen. And so if you drive into your garage with your car running and you're on the phone and you close the garage, okay, most of the combustion of that gas creates carbon dioxide, which is not great, but that usually doesn't kill you. But inefficient burning will create carbon monoxide, and just a small increase may not be good for you. All right? And so any one time, 1% of your myoglobin or hemoglobin, both of them have carbon monoxide. Hemoglobin is different than myoglobin because of this. Myoglobin is functional as a single polypeptide, one protein. Hemoglobin works together as four of the same folded protein. We actually have two versions, two alphas and two betas. They're slightly different in amino acid composition, but they have the exact same fold, and hemoglobin has four hemes inside of it. The structure was determined in 1968. Only took them 30 years to get that done, and determined that there were four polypeptides that were in a functional group. The hemes are relatively far apart from each other in that particular structure. Hemoglobin transports oxygen from the lungs to the extremities. Myoglobin binds to oxygen in the muscle. It is not a transport molecule. Both of them bind to oxygen. Both of them bind to carbon monoxide. The hemoglobin for an adult has two polypeptides, alphas and betas, to create the functional group. Fetal hemoglobin is a little bit different. It actually has three distinct types of hemoglobin throughout the developmental stage. And what we're going to find is the most prominent is two alphas and two gamma subunits. Because what we have to do is, in the fetal stage, we have to be able to take the oxygen from the mother's bloodstream, from the mother's hemoglobin, and get it to the baby's hemoglobin. We do that by making the baby's hemoglobin have more affinity for oxygen so that that transfer can happen. Once the baby is born and is able to breathe air over the developmental time, they will not produce the gamma. They will produce the beta proteins to create the adult form of hemoglobin.